Welcome back. Here we are going to discuss on the sensory structure, the sense organs of Hoplobatrachus typrinus. The sensory system includes the sense organs or the receptors, and these organs receive stimuli from the external or the internal environment, and these stimuli it is transmitted through the nerves. Okay, and in the form of impulses. So the receptors or the sense organs, they take up the stimuli, and these stimuli are then passed on through the sensory nerves in the form of impulses or the nerve impulses, which carry it from the impulses from sense organs to the central nervous system, which we have already discussed in the previous presentation. And in the central nervous system, they are actually analyzed. These impulses received are analyzed, and then accordingly, the response is being transmitted through the motor nerves. Okay, so this is how it happens. Now, each kind of stimulus, it is actually creating impulses in specific organs or the particular sense organs. Now we can see that there are different kinds of receptors or organs concerned with receiving the stimulus. In the case of Hoplobatrachus, the major ones are Tango receptors, olfactory receptors, gustatoreceptors or gustatory receptors, photoreceptors or the eyes, and statoacoustic receptors or the ears. Okay, so the eyes and ears, the structure and the functioning, we have to look into in detail. Now, regarding the first three, Tango receptors are nothing but the organs of touch. So frogs, they have numerous microscopic Tango receptors that are located under the epidermis of the skin, just beneath the skin. And these are either in the form of simple what we call networks of nerve endings scattered among cells, or as compact group of cells projecting into papillae of epidermis. They are sensitive to various types of stimuli. The Tango receptors, they pick up different kinds of stimuli, for example, touch, chemicals, temperature, humidity, light, pain, etc. We can see that apart from Tango receptors, in the case of tadpoles, they also do have lateral system, just like fishes in the aquatic medium. Tadpoles also do have aquatic life, isn't it? So for them, they also possess the lateral sensory structures other than other than Tango receptors. But adult frogs doesn't have any. Adult frogs as well as tadpoles, they do have Tango receptors. Then the tadpoles do have the lateral system along with the Tango receptors. Now, what are olfactory receptors? Olfactory receptors are organs of smell. The epithelial lining of the nasal chambers, it forms the olfactory receptors, and it contains tall olfactory receptor cells with tufts of spine cilia on the free end, that is on there are tall what you call receptor cells, and these receptor cells do have cilia which face the free end of the cell, which like the nasal chamber, while the other end or the proximal end do have nerve endings or endings of the nerve fibers attached to the inner end. Now the fibers unite to form the olfactory nerve, which carry the sensation to the brain. So here you can see simple there are long what you call olfactoreceptor cells. We can see here. Okay, so these are the olfactoreceptors, and the receptors we can find they are tall in nature or what you call elongated, and on one end they do have fine cilia. Okay, and this part actually projects into the nasal chamber. So here we can see the nasal chamber, while on the other end you can find the nerve endings. Okay, and these nerve endings, they from all the receptor cells, the nerve endings, they join together to form the auditory nerve or the first cranial nerve. Okay, so I hope it is clear. Fine. So this it is about the olfactory receptors. Now, regarding the gustatoreceptors or the gustatory receptors, we can see that it is concerned with the reception of taste. Okay, gustatoreceptors are in the form of taste buds present or located on small papillae on the tongue as well as on the floor and roof of the buccal cavity. A taste bud consists of spindle-like receptor cells, elongated spindle-like receptor cells, and these cells have very fine hair-like processes for detecting the chemicals in the food. Okay, and these receptor cells, they are innervated by nerve fibers from the seventh and the ninth cranial nerve. I hope you remember the various organs innervated by the spinal, also cranial nerves, which we have discussed when we discussed on cranial nerves. So these are the major what we call receptors. We have the Tango receptors, olfactory receptors, and the what do you call the gustatoreceptors. Okay. Now we can see this is a like yeah. The other two receptors which we already saw, the photoreceptors and statoacoustic receptors. Here we have the photoreceptors or the organs of sight, the eye. Okay, photoreceptors. They are represented by two large eyes, and each eyeball is roughly spherical. It is not like spherical as such, but it is roughly spherical, and it is placed inside this part is known as the orbit. Okay, you can see the orbit. This is the wall of the orbit. So eyeball is placed inside the orbit. Okay, and in the dorsolateral side of the head, it is protected by eyelids, and this eyeball, it is placed inside the orbit, and the movement of the eyeball, it is actually coordinated by six muscles, six extrinsic muscles. You can see here two are oblique muscles and four recti muscles. These form the muscles that coordinate the movement of the eyeball. Okay. Now the oblique muscles include the superior and inferior oblique muscles, while the recti muscles include the internal and external rectus. You can see over here internal, external, and superior and inferior rectus. So these muscles bring about the movement of the eyeball. And when we discussed about the cranial nerves, we saw that there were three different nerves which innervated or which send branches to these muscles to bring about the movement of the eyeball, isn't it? So we can see that the outer one-third exposed part of the eye here, this part, okay, this part is transparent, while the two-third part which is hidden or which is kept inside the orbit, it is opaque. Okay, and the wall of the eyeball, when we take a section of this, so this is externally, this is how it is seen, but when we take a section of the eyeball, we can see that the wall of the eyeball is composed of three consecutive layers of structures. Okay, so the outermost one is known as a sclerotic layer, the middle the choroid, and the innermost it is the retina. Okay, now the outermost one, it is a sclerotic layer. The sclerotic layer is thick protective coat made up of tough fibrous connective tissue and cartilage. Okay, and it's anterior, and you can see it continues like this. The sclerotic layer is continuing all around the eyeball, and the anteriorly, so this part is the exposed one-third part, and this part is actually the two-third concealed part, or the part which is placed inside the orbit. Okay, so the part which is exposed, this is actually anterior one. The exposed part here, you can see the sclerotic layer is transparent. Okay, so the transparent part, transparent anterior part of the sclerotic layer is what is referred as the cornea. And the cornea helps in this transparency. It is due for permitting entry of light rays into the eye. Okay. Now cornea, you can see it is in turn externally covered by another transparent layer which is known as the conjunctiva. It is actually a protective covering. Okay, it is conjunctiva. Now, a continuation of lower eyelid over here, you can see 1 minute. Okay, yeah, yeah. So the lower eyelid, it continues to form a structure known as nictitating membrane. And this nictitating membrane is stretched over the exposed part of the eyeball over here. When the what do you call, frog is in water, the water will not directly come in contact with the eyeball. Okay, for that purpose, the nictitating membrane is stretched over the eyeball. Right. So you can see there is a cornea on the anterior part, and above the cornea you have the conjunctival layer, and then even above that you can see the what we call nictitating membrane stretching out when in need. Right. So that is also referred as a third eyelid. So it's spread to cover the front part of the eye over the conjunctiva. Now both the conjunctiva as well as the nictitating membrane are lubricated. It is kept moist with the help of secretion from um, Harderian gland. You can see Harderian gland. Okay, it lubricates both the conjunctiva and the nictitating membrane. Keep it moist. Okay. Now the next layer is known as the choroid layer. You can see it is here itself. It is marked very dark. I mean, it is darkly marked over here, isn't it? So it is highly pigmented, darkly pigmented, and it is vascular in nature. That is, there are plenty of blood vessels which end up in the choroid layer. Okay, and it prevents. What happens is it absorbs all the unnecessary light entering the eyes. Okay, so this actually prevents all light except that enter through the cornea. And in front, you can see the choroid layer. It is not continuous here, isn't it? There is a space between, a gap between, and this opening is what is referred as the pupil. Okay, and around the pupil, this choroid layer, it becomes. You can see a structure here, and this structure, it is referred as the iris. Okay, this it is actually the part of the choroid which is separated from the sclerotic layer in the on the anterior part. Okay. So like in the anterior part or in the front region, choroid is separated from cornea. It forms a vertical pigmented circular disc known as iris, and this iris do have the central opening which is referred as the pupil. Okay, and this pupil, it is through this the light enter and falls on the lens. Okay, now the pupil can be reduced and like increased in the size can be reduced and increased with the help of circular and radial muscles present on the this choroid layer. Okay, and it is helps in like vision in all kinds of light intensity. Now, at the base of the iris, here you can see the choroid layer. It is actually slightly thickened, and this structure, thickened part, is known as a ciliary body. It contains ciliary muscles. Okay, and behind the pupil is present very large spherical and transparent crystalline lens. Okay, it is perfectly what you call spherical. This lens cannot change the shape in the case of frogs. Unlike in humans, we can see here it is spherical, and it is rarely this shape can be changed. Okay, and it encloses a delicate lens capsule. The It is held by in position with the help of the suspensory ligaments. Here you can see the suspensory ligaments. It arises from the ciliary body, and this helps in keeping the lens in position. Okay, now the whole structures mentioned here, it divides the inner part of the eyeball into two regions. The anterior part is the aqueous chamber. Here it is written. Okay, aqueous chamber, and this part is the vitreous chamber. Both these parts are filled with fluid-like content, and the part, aqueous chamber is filled with aqueous humor, and it is slightly more watery fluid, while the vitreous chamber is filled with jelly-like substance. It is known as a vitreous humor, and both these humors help in maintaining the form and provide proper pressure that helps in maintaining the form of the eyeball. Okay, so this is with regard to the outermost two layers. The retinal layer we will be discussing in the next presentation. Okay.